Good morning, everyone, and good afternoon for some of those who are joining, or late evening for those who are joining from afar. This is a virtual biorepository workshop. It's a very small workshop, but I think we're talking about a very large subject that many of us have grappled with for many years, and we've not been able to come up with the solution. So hopefully what we're going to do is talk about it and share some stories. I hope everyone's ready to share stories about it, and we'll hear from online and also from in the room here. All right, so we actually have, as I say, a sort of a short but intense agenda. And I hope that all of you are looking at this with an eye as to what it means for you. And we're going to do some welcome introductions. And this is the rest of and we have some examples of navigating landscape for Biorepository and so forth. And I think we have plenty of time for questions. And I just want this to be a very relaxed workshop. It's not to put anybody on the spot, but it's also you're here because there's a reason, there's an interest in it, and we want to hear what that reason is. So with that, we're going to get started. So we're going to go through a little bit of what biorepositories are right now out, what the issues and challenges are, and what some of the good things that have been done. And we have Warren Fransman from FIND, who's going to speak about the biorepository work that they have done. And it's a huge effort. And it's good to hear that, because one of the things is you can't do biorepositories alone. You need to have others to help you.
While the presentation is busy loading, thank you, May, Chu, and Judith and Julia for organizing this and for the invitation. And thank you for being such good hosts. Right. So biobanking, at least the FIND's perspective, I'm here to share a little bit of what we've learned. As I said, I'm Warren. I lead the biobanking activities at FIND. FIND, of course, is the Global Alliance for Diagnostics, and we seek to bring equitable access to reliable diagnostics to everyone around the world. Okay, so it was in response to the need for innovative and affordable tests for diseases that we find in LMICs that this organization was launched. We, of course, based in Geneva, Switzerland. We've got country offices in South Africa and Kenya and in India. And of course, our foothold is very much in LMICs, but also global. I mean, we in biobanking, we often say, well-characterized quality samples. That is what we that is what we store. That is what we always propagate and advocate for. And it is exactly these samples that that needs to be available, and not only available but accessible. I noticed that we have some ethicists or ethical folks in the group, and we all know that availability and accessibility is not necessarily the same thing. And with well-characterized samples, I mean, the associated metadata and clinical data that's needed for good information to make some decisions on with the provider data. What I do want to say is, it is quite difficult, or I would rather say it's challenging to find biomaterial of good quality and that are well characterized with the needed metadata and clinical data that we often ask for, but especially when we respond to pandemic threats, and then these samples or these diseases that are endemic to LMICs. Let me just see if I can do the pointer thing. Yeah, so I've just listed some of the phases in the test development or evaluation that process that we often we receive requests for these specific phases, but I'll speak a little bit more about that. So the biobanking activities in FIND is really based on 3 pillars. We've got the centralized biobank that's stored with a long-term partner in the US. And here we store over 500,000 samples across the FIND disease spectrum. Undoubtedly, TB samples takes up most of our space. But as you can see, we aim to provide fit for purpose collections and store these. And most of these come from low to middle income countries and endemic areas. We aim to support, well, not support, but really rectify and address health inequalities in this way. And we provide samples. And I mean, I just need to note that our sample access process is well documented on our website. We obviously have a SBRC, a specimen bank review committee, and each and every request coming into our website, there's a material request form and a material transfer agreement, which is a legal document that needs to be reviewed and signed by both the requester and FIND, who's the custodian or custodians of the samples. And once this document is, signed, and the review committee has provided a go-ahead to share the samples then get shipped. We make use of Open Specimen, a database to track our samples for obvious reasons. The map shows just, let me just see where can I press again. The map there, I just wanted to highlight, where our samples are currently coming from. This map has been, it's 2019, so we've added a couple of more sites to our partner sites or network. So we are continuously reaching out to sites and partners to set up collections for biobanking and test evaluation. The other 2 pillars that the biobanking activities at FIND is based on is around the FIND integrated biobank. And this really is a disease agnostic. It's a decentralized approach to biobanking. And why this is very exciting is, we all know about how difficult it is or challenging at times to export samples, depending on the country. And we've since at the end of 2020, beginning 2021, we've set up what we call network sites, and we really, then equip the sites to not only collect samples but store it in country. And then the idea would then be, test kits would be shipped to the PI, to the lab, and they would do the in-country, or they would do the test evaluations in country. A portion of the samples invariably gets shipped to a partner labs, or just to do EQA panels or lab concordancy studies. The third leg of our operations is the DX Connect Virtual Biobank. This is an open access platform where FIND plays the connecting role. Basically, what we're asking is, anyone and everyone that has a collection of samples, a biobank, a network of biobanks, come to our website. We no longer want accessibility or availability of samples to be a hurdle for test development to occur. So we're saying, come and register your collection, and we make it visible to sample users, whether they're from industry, academia, or commercial entities. All our efforts go towards test development and moving that forward. I'll speak a little bit about the virtual biobank at the end of my presentation. What I do want to just maybe for the next 5 minutes focus on is the storing of samples in country. As I said, we've got this network of sites. We just signed with a group in the DRC for a monkeypox collection, but essentially we've got in South Africa, in fact, yeah, at UCT, we've got a fine integrated biobank network site. We've got one in Peru, Moldova, and Nigeria. What is really interesting, or what is really gives me peace of mind about this setup, is we have about 65 work instructions, policies, and forms that's part of the defined quality management system that we share with the site. So when you get a sputum sample from Nigeria or Moldova or South Africa, it has been collected, processed, quality controlled, and stored in the very same way, and it will be distributed following the same SOPs, which brings me to the point. So with fine distribution of samples is key. We go at, we put in every effort to make sure that we distribute our samples to anyone and everyone that needs it. Obviously, in as long as the reason for the request is aligned with what FIND wants to reach in terms of our goals. Why I have this slide on, so when you go to our website, we've got handling fees, right, on our website. And we can probably speak about the sustainability of a biobank and operations for hours. So we've got what we call a handling fee, and this is really just to recoup some of the cost. And we all know it's a very, very expensive exercise to set up biobanks, to do collections, ethically sound collections at least, and to store these and to ensure that these samples remain intact, right? And so when you go to our website, all of our diseases have a different handling fee. It's a handling fee structure that has been approved by our board in 2019. But our COVID collection, so we've got over 100,000 COVID samples, bloods, and swabs, and we're making this available without any handling fee. We do obviously ask that the requester pay for the shipping. The monkeypox collection is soon to start February 2023. We are busy, getting ethics approval and contracts, and how long that takes, and procurement, of course. Right. So across the fine disease spectrums, we've got, a substantially large representation of samples, and our sample types are also what we perceive as what has been requested by industry partners at the time. The one thing that I do want to say is I remember when we started out with the COVID collections, we at some point only collected swabs and VTMs, and at some point everyone said, no, wait, we need dry swabs. So back to ethics, and everyone, well, we were then collecting dry swabs because of the VTM and the manufacturers' media that we needed to do antigen test verification on, and it doesn't mix. But I want to say, so after months of collecting dry swabs, the one thing that I would like to maybe discuss in this group is how do we get the timing right from when it is needed and when we actually have it in our freezer? Because now we have 100,000 samples. It was at the time requested that we collect these sample types in these conditions stored. But now all of a sudden everyone moved on, or they have their own samples. I'd like to get your perspective on how do we get the timing right. And I think this is my second last slide, if it's not the last. So basically the DXConnect Virtual Biobank open access platform is to, number one, to showcase what is available in in freezers everywhere. As I said, FIND plays the connecting role, so we don't get involved in IP publication rights or even the MTA and MRF review and signatures for the specific platform. All we're doing here is saying, look your world, manufacturers, academic partners, industry partners, here are the samples. If you need them, please connect with the sample owner. And the specific project was funded by the Gates Foundation, and we aim to populate this platform to such an extent that sample accessibility or sample visibility should no longer be an issue, not in 2022. I'd like to hear your thoughts on that. So it is a collaborative approach where we're saying sample owners, sample users, here's the platform, tell us what you need, tell us what you have, and let's make test development happen. Thank you very much. I'll take questions.
Oh, okay. My name is Mercury Shitindo. The current interim chair at Africa Bioethics Network, and we have recently just established an African journal for bioethics. So my question is around that area. First and foremost, that's a very good initiative, but I'm wondering what are you going to do, or what is your plan on including the ethics bit of it, like making the samples ethical, like at which point will the people who have given the samples be involved, and what is the consideration for future use for the samples, if it's going to be in a repository, what are the ethical considerations? So I don't know if you had already answered that. I came in a bit late, but maybe you can address that. Thank you.
Thank you for your question. I'm assuming you're referring to the DX Connect Virtual Biobank platform, right? So just as a reminder, so we've got our central biobank where we have the material request form and the MTA that needs to be signed, and all of our collections in that platform, in that space, has the necessary ethics approval, and we store that because we set up these collections. It's FIND own samples. So for the DX Connect Virtual Biobank platform that you're referring to, where FIND is merely playing the connecting role between owner and user, the owners rest on those two parties to make sure that there's consents, informed consent for those collections, and as I said, the MTA MRF needs to be signed by those two parties. The only role that FIND is playing in this regard is increasing visibility of collections. But I understand your question, and it is something that needs to be discussed by those two parties. Over. Thank you.
Jen, from Cambridge, I said I'm working on an ethical study around biobanking. My question is similar to what she was saying. If you say you consented these participants, did you consent them, telling them that you are going to make this sample available? Okay, what can I say? Yeah, like, did you say that you are going to make these samples available to everyone to see that the samples there, or you only consented for the sample storage and sharing, or how did you do the consenting? If you didn't, maybe say these samples will be made virtually, everyone will be able to see what you have. Then what's the next step to be taken before you make these samples available to everyone, or you make them everyone to see that this is what we have?
Yeah. Thank you for that question. So the DX Connect Virtual Biobank platform is, when we advertise, for lack of a better word, the samples, it's not at sample level. It's merely at at organizational level, and it gives sample type information and storage conditions. But the key thing here is for, as I said, the user, a potential user of these samples or requester to connect with the owner and to sort out, the ethical concerns and legal concerns. But to address your question, when it comes to our centralized storage and the collection that we, own and are custodians of, we from time to time have an ethicist, come in and advise us on, going forward, because it is true what you're saying. 2007, we had no idea that we would have a virtual biobank and that we would increase the visibility of our collections for test development. So it is something that we need to either address when we do consenting, and and the ethics committees are very involved in this. So just as an example, we've got organizations coming to us and saying, look here, I want to register my collection, but I need to make sure from local ethics that this would be okay, because at the time of consent, this was never planned. So your comment is taken to heart, and it's a very good forward-thinking suggestion. Thank you.
Well, thank you for your presentation. My question still revolves around the same in terms of participant consent. So most times we tell them to consent for samples, this sample storage for long-term storage,
Yep.
and we give a time frame as to when the samples will be destroyed.
Yeah.
With the virtual, do you have an oversight body if you're to link the user and the site as to when or how you'll oversee if you've said the samples will be stored only for 10 years, so that we don't have the site using or exiting without ethical approval?
Yeah.
Yeah.
I've really opened a can of worms there. I'm in agreement with what you're saying, and so once again, for our find own samples, we in fact do a 2-year review of, is the language in our consent form still valid, and does it still extend permission for what we're trying to do. So as an example, we have requesters coming in saying, we want to use your samples for research purposes, right? And then we have to go check for that collection. If this requester wants samples from this collection, did we say, does the information or the language in the informed consent, does it permit research, general research, or is it for test development and LMICs? Because we've got collections that's specifically for that. So to answer your question, we are very much cognizant of the language that's captured within the consents for our collections. Hence is why I'm saying that, when we do make that connection between a owner and a collector of samples, they need to, pull out that consent form for that specific collection, for those samples being requested, and ensure that they comply with, the permission extended. That is all I can say to that. I know it's not a perfect answer, but thank you very much.
A recommendation would be to have policies in place, develop guidelines, policies, and maybe SOPs that guide how samples or informed consent should be taken, and then maybe give training maybe to the people who intend to use it. And this should also be shared with some of the ethics committees who approve such. So yeah. That's all.
Thank you. Thank you for your time.
So now I have the privilege of introducing Nikos Vasilakis, who will talk to us about regulatory and compliance challenges for the maintenance of biorepositories. As Nico said, he's a tenured professor and vice chair of research for the Department of Pathology at the University of Texas Medical Branch, and a colleague in the CREED network, someone with whom I have broken bread and learned a lot. So Nikos, I'll let you take over.
Thank you for Can you hear me? Thank you for the invite and to be able to bring to you our US-centric concerns about regulatory and compliance challenges, and I will give you some examples of that as we move forward. So I also represent the World Reference Collection of Emerging Viruses and Arboviruses, which is located at the University of Texas Medical Branch. Just for your own information, we have over 10,000, actually over 13,000 virus samples belonging in 21 families, 776 species. We also have a collection of antibodies, mainly mouse hyperimmune sera, monoclonal antibodies, and a collection of human convalescent sera through the collaboration with the Global Virus Network. And also we produce and supply antigens, infectious cDNA clones, and as well as cell lines. So under this capacity, I will give you a brief overview of what we consider the challenges, and particularly pay attention to two often underrepresented concern, that is of select agents. So I don't want to take Tony's breath or thunder away, but this is a brief overview of what biorepository operations are. And what I would also like to talk about you is towards the last part, the shipping requirements and the permit landscape, and how that covers both institutional, national, and international boundaries, and of course, some of the regulatory compliance. Okay. So in terms of the international compliance, the guiding light for that is the Nagoya Protocol, which was developed to provide a fair and equitable sharing of benefits arising from the utilization of those samples, had it be sera, genetic resources, viruses, you name it. There are country-specific regulations that also supplement the Nagoya Protocol, such as various ministries of health, agriculture, agents, and many others. There is the World Organization of Animal Health, the International Plant Protection of Convention, International Air Transport Association, to name a few. And of course, the Convention of International Trade of in Endangered Species. And I will share with you a couple of horror stories that we had for that. But mostly, I'm going to talk to you about the national aspects, since this is something that I'm very familiar with, and especially with the permitting agencies in the US, and these are listed up there, and also the select agents. But a lot of the, I would say, the bottlenecks that we are faced, besides all of those, are the institutional regulations, and mostly of the availability to have in an expedited matter a material transfer agreement, so the exchange of materials can start from the biorepository to the requester. Oops. Okay, so let's start with the permitting agencies. The CDC is the major agency that regulates the various aspects of those agents. And especially it is important that anytime that you want to bring an infectious agent or antibody or any of those vectors that including mosquito sticks and so on, you need to have a permit. And that permit, it takes approximately 6 to 8 months. You cannot have a global permit. Some of it, they are highly specific, and the regulations become extremely, extremely strenuous when we're talking about high infectious agents, and especially select agents, or from the agricultural point of view, sera that may be coming from ungulates or samples from specific animals or ticks from areas that they have specific diseases or vectors of veterinary importance. So this is one of the agencies that we need to be worried about. And most importantly is I would like you to focus on the penultimate bullet, that the importer is legally responsible to ensure that the import permits are in place and put together in the package and so on. And most importantly, comply with all the regulations. The next one would be, again, in terms of the select agents. Now, everything that I showed you before does not apply virtually. You have an additional paperwork on top of that. So in terms of that, you need to be able to, when we're talking about select agents, first of all, if you're sending an antigen from a select agent and so on, you need to make sure that it's properly inactivated. You have all the documented procedures that indeed indicate that it is an inactivated sample. And all of this came of an accident. Usually all of those regulations come out of an accident. And the select agents regulation, which I'm going to talk to you about a little bit later on, they were all derived, actually, that particular one derived from an accidental shipment of Bacillus anthracis that they claimed that it was inactivated, but in reality was live. So there was no regulation at the time. So it was necessary to develop the regulation to cover ourselves. On the next one, in terms of what you're bringing in, also the United States Animal and Plant Health Inspection Service also requires a permit. And this is mostly from agricultural biological agents. And as you can see, it mainly affects the veterinary landscape. So everything that it comes to that is basically covered, from animal sera to tissues, blood, RNA, DNA extracts, as well as hormones, enzymes, and so on. So if a collaborator in Kenya wants to send us a sample from a cow, you need to have those permits, and also whatever you send, you need to certify that it's completely inactivated and it doesn't have an infectious agent. U.S. Fish and Wildlife also deals with wildlife specimens, and the CITES Convention on International Trade of Endangered Species. This is actually a particular peculiarity of the United States system. So we all agree that CITES and protection of wildlife is essential for the well-being of the ecosystem, society, the world, and so on. There is no, I would say, argument in there. So in the United States, any cell line that is derived from an endangered species, doesn't matter how long it has been in place, it requires a CITES permit. So, for example, if an investigator in South Africa asks me to provide him a sample of chikungunya, for example, and that sample has been grown on virus cells, I am required to obtain a CITES permit, because it is illegal to send anything like that that has grown on virus outside of the United States without a CITES. Now, in case of an outbreak, there are a lot of complications of that, because to obtain that permit in the United States, it takes about 6 months. And the amount of paperwork, it's insane. So if an investigator in South Africa requests a sample to develop antigens or other diagnostic tools, has to wait all this time, because it is impossible for me to send that specimen out of the United States. And if I do without a permit, there are extreme fines and imprisonment. So, but if I ask somebody from the biorepository in France to send you one, that will not be the issue because the European Union does not require a CITES permit for anything that has grown on virus. And this is an overinterpretation of the rules and regulations, the legitimate rules and regulations of the CITES. This cell line was developed back in the 40s and 50s, so it should have been grandfathered in, but apparently it is not. So this is something that is a major, major roadblock in sending samples, free sample exchange from biorepositories to requesters and so on. But I would like to talk to you in the next few slides about the select agent regulation. Most of it was, it initiated back in '95 when somebody in Ohio asked from ATCC to obtain some samples of Yersinia pestis with no paperwork. And then we realized that this is a no-no. That should not have been done because that investigator had nefarious purposes in his mind. So there were a bunch of regulations that they started that beefed up those regulations and the exchange of materials and what should constitute as a select agent. And just to give you an example, this is exactly what is a select agent. There are biological agents and toxins that they have the potential to pose a significant public health threat, not only to the public, the animal, the planet, of course, or have a tremendous economic risks because the unknown, actually the elephant in the room, anything that it has to do with infectious disease or samples, it's all about economics. And I'll tell you a little bit about that in a little bit. So there are a bunch of agencies that they control that. And along those lines, here are the number of specimens that they fall under the select agent regulation. Most of them, they are tier 1, that means hemorrhagic fevers, Yersinia pestis, anthrax, and several others that have been for a long period of time responsible for major threats. So I'm not gonna, this is just to give you an overview of what is going on about the degree of paperwork that is required. Not only in terms of how long you need to keep this paperwork, what are the institutional, national, and governmental regulations that they cover that, but also the training that it's required. And this is a major problem, especially when we are talking about exchange of those materials. And I will give you an example. Guanarito virus was isolated in Venezuela back in the 70s. That sample was given to CDC for characterization, et cetera, et cetera. Now, this virus is a level 4 agent. That means that you have the highest level of biocontainment and security to be able to safely work with that. When an investigator in Venezuela in the 90s requested the sample back, we could not give it back to the investigator because Venezuela at the time, and still now, does not have the biocontainment requirements to deal with the agent. And that, again, especially for the CRED network that we're dealing with most of the time, select agents is a big problem because there is no infrastructure anywhere to be able to successfully and safely deal with select agents. That is more complicated if the work that is being done in situ anywhere in the world is funded by the National Institutes of Health. If it is, then you have to abide with those rules. And you cannot send and receive specimens that are under the select agent category unless the requirements are in situ. And some of them, they are in here. What does it mean? Risk assessment of the personnel. Everybody goes under extensive background check. credential checks, medical and mental health assessment. And that's not the end of it. This is just to get into the system to be able to work with that. Once you are into the system, then there is an ongoing assessment as you work forward. There is a criminal background check every year. They evaluate the credit, not only your financial credit worthiness, but also your moral and integrity and trustworthiness on a real basis. So, for example, if I have an investigator in my program that it has some serious issues that, either family issues or any others, and that he or she is not fully able to have his or her attention and work effectively, I immediately pull that person under the program because he or she could not work safely with those agents. Because here are the requirements, both on the facility accountability, not only, let's go back, material accountability. This is what you need to have. You have to have robust agent inventories, animal inventories, autoclave records, you have decontamination records. You have to follow the agent from inception, when you generate it, how you use it, and then how you discard it, and documenting that is happening. In the terms of transportation and security, you have to have chain of custody, which increases significantly the amount of cost. So, for example, if Tony in Duke wants a select agent and/or any of those agents from me, it's a minimum of $5,000 to send one vial containing 500 microliters of that agent. And that vial, it will be the only package in the back of a truck going from southern Texas all the way to the Carolinas where Tony is. $5,000 for an item like that. And usually it requires 2 drivers, police escort, and so on. So facility security. For some of you that may have come at UTMB, especially in the biorepository facility, is like getting into a military base. There is airport security, screening, there is advanced screening of the individual who comes, background check, and so on. So there are a lot of requirements with that. And of course, the personnel liability. Now, all this together increase the cost, not only the maintenance, but also the ability to share those agents. So there are only a handful of facilities that can work safely with the select agents simply because of the cost. I mentioned before, the cost of non-compliance are significant. $500,000 fine and imprisonment of several years. And this is something that is serious. The CDC and other regulatory agencies, they have yearly audits of all the facilities and the investigators who are working with those agents. So anytime, and those visits that they're coming to the facility are completely unannounced. Nobody knows when they're coming. So when you're coming, they're coming, you spend a week with them, they go every single record, spot checks in the freezers, opening the boxes, which are sealed, by the way, and say, okay, tell me what you have in here. And if the records don't match what you have in the box and what the records say, you're in big trouble. So where we go from here? And this is something that we need to discuss after tea. We need to have a pragmatic overhaul of the regulatory landscape. Currently, there is an overregulation, which is it's a true barrier to access and sharing of those materials. And especially when it comes to well-meaning protocols like the Nagoya Protocol, the ultimate result is that they have the opposite of what the founders intended. It is impossible, it makes impossible to share, but also there are numerous bureaucratic obstacles that make impossible to share any of those agents. And of course, there is the affordability. We need to make a distinction between academic research and versus commercial development. And this is something that a lot of times it is not discussed, because this is the elephant in the group. Most of the samples are coming from LMIC countries, and the true discussion has to be done. If an LMIC country provides a sample, what the sample is going to be used for. Is it going to generate a vaccine? Is it going to generate a commercial kit? And if that is to happen, what is the responsibility of the developer to share equitably the profits of that? So there are a lot of discussions that if we need to move forward, those discussions, they have to be made on an equal basis with a frank discussion and understanding the limitations or the sensitivities of all parties concerned. And then we need to have a framework of principles for outbreak responses. Zika, dengue, monkeypox, and so on, they have a lot of those limitations. We need to be able to address the stakeholder sensitivities. We need to have consultation with those stakeholders and the end users. We need, and this is important, we need to have equity sharing of those materials. And importantly, we need to have local capacity building and engagement at the local level if we are to be ever successful. So we have a lot to discuss in the chat. And these are the acknowledgements that I would like to make, especially at the WRCIVA, the Department of Biosafety at UTMB, that keep us safe and provide us all the regulations that we need to abide. Tony and Megan, that we had many discussions, and of course, May, Judith, and Julia for very fruitful discussions where we go from here all this time and how we can make it happen, and of course, the funding. And with that, I'll stop. Thank you.
So it's a very mind-boggling set of slides.
Yes.
Right? So this is May Chu. And I have to say, I've been on the other side of some of this, right? Because I was at CDC and I was responsible for compliance to the select agents. So many of these painful stories I have heard. And I would agree that things should be looked at much more practically. The 1995 incident that you mentioned, I was a recipient of that phone call from the FBI. So I remember that very well. But I think the thing that, sort of the snide remark would be, don't do business with the US because they're not available for all these reasons. But however, the takeaway ought to be that some of these rules and regulations were from mistakes and need to be looked at globally as to what risks we're putting people at and the populations at for these kinds of activities. And that there needs to be a frank and open discussion and also a change in paradigm shift. So I think the question is, where do you think we need to go? Are any of these regulations in the US that you discuss applicable globally? If they receive NIH funds, they are already compliant, but other countries don't have that at the moment. And do you think we need to have some kind of security or check or balance, and to what extent that could be?
Yeah, thank you for this. Biosecurity is an extremely important aspect of what we do. I think the United States is putting, is leading the pack. But those regulations about biosecurity and so on, they need to be developed in situ to be able to address the sensitivities of its country. I think it's a mistake we have many examples in various aspects of life that trying to impose a set of rules and regulations in an environment that it has different cultural norms, economic conditions, et cetera, it does not work. It's not practical. So I think those rules, they can serve as a baseline, as a foundation that can be tweaked based on the sensitivities of its country. And also they have to make sense within the country. And that is going to take some time. I don't think we're ready yet. We need to be able to sit on a table with no conditions and be able to discuss and all come to a consensus and understands each other's, I would say, sensitivities. This is critical. This is the only way it will work. But it is due now that we need to have some global kind of setting of regulations that they are uniform across the spectrum in a way that makes sense.
Thank you for outlining some of the challenges we are facing. I want to challenge you. You're saying that we need to. I think we need to change that statement to how do we start this? How do we initiate this conversation? And I'm hoping that by the end of today, we are going to say, okay, we can have this first discussion and then we see where that takes us.
Yeah, absolutely. We need to start somewhere.
A slight reaction, and I agree with you. But one of the challenges I think that we all face is that a lot of this regulatory landscape it's being created by people who are not experts in the field, and we don't always have the ear of the legislators that are actually putting these regulations in place. The laws in the US, the laws in the EU, the laws in Kenya, the laws in South Africa, they're similar, but they're different. And so it's difficult to come up at the moment with any kind of universal anything because everybody views these questions slightly differently. So I think we can come up with a number of recommendations for, how to deal with it. And we can certainly give advice in specific situations, but it's really hard to come up with anything that's universal right now because we would have to get all the legislators to agree. Mm-hmm. And I'm sure all of us recognize the challenge of, the US, you guys get the news as much as we do. It's a glorious disaster. I think it's challenging to get all of the laws to align so that we can do things that are more universal or more global, if you will. But that doesn't mean we shouldn't try. So I agree with you. It's how do we start that conversation? And at some point, how do we engage the people who are making those rules and get them to actually understand what the challenge is? For example, you take a sample from a location that's essentially swimming in that disease and say, I can't send it back because it's a risk. A risk to who? Like, what in the world? Where is that coming from? And it's fundamentally because the US is an incredibly risk-averse place because, the last thing they want to see is you sent that back and then it got, diverted to something and then, well, the US caused that, right? Yeah. Nobody wants to deal with that. But that doesn't make it the right approach. Yeah. And getting legislators to understand that takes a lot of time. And they don't have a lot of time. They have like 30 seconds and then they're on the next thing. So.
Yeah, thank you, Tony, for bringing that up. It is a challenge. Many of those laws are written by the regulations are written by lawyers with little input from stakeholders, and the lawyers don't understand what a virus or antigen or antibody is. So if you say, for example, a last antigen, they say, oh yeah, I'm going to die, but it's an antigen is not infectious. But that conversation, that advocacy needs to start. And I'll give you the example, what Tony mentioned before, that dengue viruses, because dengue hemorrhagic virus, the old nomenclature of that, dengue was in the Department of Commerce list of export control viruses. So if you, for example, have given me a virus, dengue virus from Kenya, to me in the United States, and then you asked for it back, I would have to get a Department of Commerce export license to send it back to you. And this is another 6 months of paperwork for somewhere that the virus is basically swimming. You know, you can get it anywhere. So it took advocacy on our part that we educated some of the people at the Department of Commerce. So now dengue is out of that list. Oropus is also downgraded, and West Nile is also downgraded in terms of biosafety. So it takes a lot of effort and discussion to do that, but we need to start somewhere. And each one of us were the best ambassadors for that. Not to get frustrated, to engage, and talk. And many of us, we don't like to talk. We don't like to be in conversations because we are busy with many other things, and you know, we make assumptions. So I would urge all of us, just start the conversation, talk to each other. Thank you.
I don't know how many of you, yeah, I know. I don't know how many of you realize this, but there was a discussion about making SARS-CoV-2 a select agent, right? What sense does that make? And yet that was an active discussion of, should we take a virus that has circled the globe and turn it into a select agent so we can't actually work on it? So that's the kind of thing we're doing.
So I have a rejoinder to that, then we'll go to the next part, which will shorten greatly to keep on time. And that is, I think, of all the rules for biosecurity and the select agents, I think we started on the wrong footing. And I have to confess, I was at the beginning part of it, 1995, 1996, 2001, all those places, engagement as part of CDC. I think we always did it wrong. And that is, and I can say that because I'm no longer working at CDC, is that it's a process and the mindset of what you're going to use and how you're going to use it, the dual use threat of it, if you will. It's not the agent, it's what you do with it and the process by which you, quote, as they say, weaponize it. That is what should be regulated, not the agents and not the samples. And I've been fighting that for a number of times. So we're going to have to talk about it more at another time and figure out the advocacy part of that, because we can make changes, but it's going to take that kind of argument and ethicists to also talk about the mindset of using something for the wrong reasons, right? And we have to make that strong argument about the correct ethics. So my opinion over. So let's go to the next slide, set of slides. This is to follow up on the last two talks, and thank you both, Warren and Nicholas, for raising some of the things that have been really good and some of the things that are barriers. And so we just want to have a short section of time to talk a bit about what are some of the barriers and benefits of a biorepository. And this is sort of what we call open microphone session. And and I think we've already talked a bit about the biggest barriers to participation. Are there anyone that would like to add to this? Any other kind of barriers? I think Nikos mentioned Nagoya Protocol. And at some point, we can also discuss a bit about Nagoya Protocol, because it's something that a lot of people don't understand what it is, but your government likely has signed to comply with the Nagoya Protocol, but they have to also decide for the country what the benefits are. And that's still a barrier, and we haven't really touched on that very much. And some of the things that we talk about, barriers to solutions, I think, the select agents and the biosecurity and the secure and safe use and the for the purpose of benefits and good use of pathogens and samples and materials need to be discussed in the context of biorepository, because it's crucial to us so that we understand when you receive that sample, let's say through the find exchange, you know exactly what your responsibilities are. I think it's very, very important. And then, the other things are, the barriers and benefits. It's just simple for a site, a site that has samples that may want to share it or may want to store them and share them. Then what are some of the cold chain needs? That becomes a barrier, and that I think we should also, through our work, and you'll hear shortly from Judith, is I think having a costing tool so that you can, at your site, at your level, begin to think about how much does it cost to actually carry out a biorepository work. How do you maintain it and how to serve it? Because you have to be able to communicate with your senior people and your colleagues about what it costs and what the people costs are, the monetary costs are, and the risk and the cost of the security and safety, as Nikos has mentioned, is quite important. And so, and the other thing that might be tools that you want to help with is, for all the things we talked about, we need to have, and I don't know, Warren, if FIND has checklists or things where you can say, I've done this, and this is correct. Now I can go forward. The ethics part, have I done my due diligence on certain things? So these are some of the things that we want to think about as we go through the rest of today about what the barriers and the benefits are for operating a biorepository. In our case, we're talking about virtual, where the local conservation is done at local sites and custodial sites, and that these specimens are precious and they are not yours per se. They're owned by your government or by community, and you are really the temporary custodian of it, right? Because the thinking is, a lot of scientists say, these are my samples. These are my legacy samples. I collected them all these years of my research. They're mine. And, what we have to do is inform people that it's not theirs. They have permission to temporarily hold them. And as they pass through and move on, other people take on the custody of these, and they have to be trained. They have to understand their responsibilities. These are things that I think, as a group working together like this, we can articulate those benefits, and we can articulate some of the tools that you can use, and you can move from there. And we're not stuck with not being able to share. So these are all the comments. I just want to summarize some of the things that Warren and Nikos mentioned. And did I miss anything? Any part? Okay. So take a deep breath. We're going to pivot a little bit, okay? And if you want to, you can stand up a bit and walk, stretch a bit, but you're not out of here till 10 o'clock, because that's when the tea break is. So I'm going to introduce the next speaker. It's a colleague of mine for many years, and I think we decided, until we got to Cape Town, it had been 7 or 8 years since we last saw each other. COVID has done an interesting turn on many relationships, and we used to work together at the CDC. And what we did was, with the virtual biorepository work that we've done, that Judith is going to share with you, is about what we did was, we went to a group of experts, about 420 some experts outside that are interested in biorepository retainment of samples, and ask for the opinion. What should we be doing if we're going to do a grassroots effort? What are our responsibilities? Because we all know it. We all say it. You've heard it already, but what actually matters to you? And what we wanted to do was to hear from them and find out. Okay. And before I have Judith start, are there any questions online? Okay. No? Okay, so Dr. Geary, I want to introduce, she led the discovery and research for biologics and antibody-based therapeutics in several biotechnology companies. She's quite experienced with the biorepository guidance. She was part of the ISPR group that you originally put together the guidance for the biorepository operations. And she joined CDC, where she and I had the fortune to work together for a number of years, where she oversaw the CDC biorepository. And and since then, we've been fortunate to work together as a part of a team, thinking through what biorepositories should mean from the grassroots level, because there are large formed biorepositories like at UTMB, at CDC, here in South Africa and elsewhere. And those are, funded, operationalized entities, but there are a lot of opportunities for smaller groups, as you hear from Warren, across the world. And what we wanted to find out was, what's really going on in people's minds and what they want, and what are some of the priorities that we want to follow up on, because there's many things that we can do, but what should we do first? And so we went through a consultation on a Delphi process. And so Judith is going to share with you the early consensus results of that.
Thank you, May, for the introduction. Excuse me. Can you hear me? Great, thank you. Okay, so I'm going to start out by acknowledging all the people we work with. I think you have already met most of us from the University of Colorado School of Public Health. It is May 2. Thomas Yanish, who's not here, are co-leading this effort. Julia Poggi is our project manager. And we have a couple of additional helpers. Wanted to point out Dr. Amy Price from Oxford University, who is an expert on this survey process that we will describe. Part of our funding is also from this project that is supported by the EU. It's called Reconciliation of Cohort Data in Infectious Diseases, or RECODED. And I wanted to then mention also our colleagues from the Center for Research in Emerging and Infectious Diseases, so CREED. You've already heard about Dr. Tony Moody, Megan Everett couldn't make it here, and Nikos, who you already met. As I think Warren has introduced very well, the timely access is kind of the key, the timely being a keyword. The access to well-characterized quality specimens and associated data remains still a critical unmet need every time there is a new outbreak. And this concept of a virtual biorepository came about in conversations with a lot of people. The idea is to facilitate broad access, and with the emphasis, as May has already mentioned, on locally owned and governed specimens. The initial focus for this virtual biorepository system that we'll talk about today is on specimens specifically for serology from immunological approaches, whether it's diagnostic tests, vaccines, various panels, reference panels. The guiding principles for the virtual biorepository are the same that have already been mentioned for FIND, as well as the ethical collection and use of specimens. And many of you are interested in that part of it. And then creating a trusted system with benefit sharing, and that's sustainable. And our efforts will be in alignment with other ongoing biorepository initiatives like the BioHub that the World Health Organization is doing, FIND, and many others. This is how this started in 2019. In the Zika outbreak, there was a symposium that was done together with FIND about this whole issue of specimen access to specimens and what are the challenges. And we followed up with a couple of workshops with the idea of, as May has already introduced, of getting really feedback from stakeholders about what the really needs are, what are the challenges, what are barriers, and what are solutions to some of these barriers. And we have conducted surveys with and interviews also with commercial partners, those who are developing the tests, and what their needs are and what are the barriers are. So trying to find out really what are the main issues and how we can approach them, and not from a top-down approach that here is a system and we'll build it and come and use it. It is more from a grassroots approach on what are the needs, what people see as the main needs. I already mentioned we have used two workshops that we've done together with the Global Health Network. We have collected information, and the next step was to try to identify what are the priorities to actually operationalize, how to reduce this to practice and actually start this virtual biorepository. And for that, we have employed the Delphi process, which is a consensus building survey process. This is sort of what the basics of the Delphi process are. You identify your main issues, you would do your research, then you do 2 or more rounds of surveys to arrive at consensus, and then kind of go to the next step of actually operationalizing it. So we have done already, as I mentioned, workshops. We have formulated the questions we wanted to put in there, and then piloted this Delphi survey. We finished the first round of it, and next we are going to do a second round, and we have an advisory panel that will help us kind of collect all this data and arrive at what is the consensus and what are the key priorities. And then the most important thing we want to do is recruit in this conference 10 sites that are willing to start, and so that we can actually do a pilot and identify what are the things we need to do to improve the process. So this was the method. I think we reached out with Julia's help to over 236 participants, and we created this questionnaire that had about 34 sections in it. And then people were asked to rank order their answers, from not important to possibly moderately important to very important and the most important. And we have, as I mentioned, completed so far the first round. So one of the questions we asked is to just get an idea of what type of biorepositories people are thinking about as being most useful. Again, keep in mind that we are talking here primarily about, not about collection of pathogens, but collection of samples. And one of interesting result we found out that a lot of people thought that maybe up to not a more distributed system rather than a centralized biorepository. May mention like the biorepository, the CDC, that's a central biorepository. All the specimens from all the CDC labs go there. The idea here is, what do people think about a more distributed system, especially a federated system of locally owned specimens and locally governed specimens? And it seemed that most people were interested more in that, in a federated system than necessarily the centralized model. Again, not biorepositories have a lot of different purposes, and one size doesn't fit all. There may be also some possibilities of some kind of hybrid or combined system. But this is the results we got from the survey. Who were the survey participants? They were from many different places, quite a few from North America. What we hope in this meeting that maybe we can ship this and more from Africa, and other places will be, LMICs will be involved. A lot of the people who are living in the US, however, and in the countries that are shown here work in LMICs. And you can see about over 20% in Africa and in about 14% here from South America, about 15% from Asia. So their work and it is distributed there. The respondents were working different types of organizations and have different roles in these organizations. So the first question we asked, again, about for specimens, specifically for immunological type applications, what are the type of specimens that people think are most important? And this shows only what people have marked as being the most important, either very or most important. And of course, for immunological purposes, blood would be an important, peripheral blood molecular cells were ranked high, Ig fractions and so on. Nasal swabs, although this is again intended in a pathogen agnostic way, we are thinking very much based on what our most recent experience has been, of course, with COVID. In the comments, people mentioned various other types of specimens that would be useful, in particular tissue specimens for immunohistochemistry and other uses, and other body fluids, and that would obviously depend on the type of pathogen. The type of specimens that are needed are longitudinal serial samples from confirmed cases, samples taken before and after vaccination, specimens from regions with a known endemic disease, and of course controls as well. One of the vision for this virtual biorepository system to start with is that people will put aside, people are interested in joining this, will put aside a set of specimens, high volumes, 10 milliliters if possible, and that this will be well-characterized quality specimens. So it's not the entire biorepository, but a set of samples that people will put aside. And the question we asked are how important are these really having high volume samples for different uses? Of course, it depends on the type of use, but if you look at the yellow and the green are sort of the very and extremely important for reference material, for calibration. Obviously, for that, it's very important to have some of these type of characterized high volume samples. When you're looking at seroprevalence studies, that may be less important. So it depends on the use, but that everybody found that it would be helpful to have this kind of collections that are high volume samples, well characterized samples with associated data, of course. And for research, it sort of would depend. For diagnostic tests, again, it is very important then to have these high volume samples. The samples without data are not useful for very much. And so the question was, would people think, what do people think about having a minimal set of data that will accompany the specimens? What would those minimal type of minimal data requirements be like? And of course, you need to know, it is very important that you know where the samples came from, what type of samples they are, how they were handled. This is the quality part, how they were handled, how they were collected, how they were stored. And then if they've undergone any freeze-thaw cycles, and that again depends on the use of what you are going to use the samples for. That has to do whether it had happened during shipment or whether you had some breakdown in your facility. The fit for use concept has already been introduced before. There might be some uses for which all of these are extremely important, and there are other uses for which maybe you could use the samples. What are the criteria for a tiered kind of system of ranking the specimens? And of course, you have to have they have to be accessible. They have to have clinical data associated with them, the demographic data, clinical data, test results. You need to know something about the storage conditions and how they were handled, what tests were performed, and something about the volume. So this is sort of a list of what would be kind of a way of ranking specimens for different uses. This is interesting if asked how people would consider themselves who participated in the survey. Are they users of specimens, or they think of themselves as contributors? And most people have think of themselves as both users of specimens and contributors. And that is, I think, kind of colors how they answer different questions. Before I go to the barriers, we also asked the question about all the COVID collections that are around. And there are a large number of biorepositories that collected COVID-19 samples. And when are they going to do that? Maybe if this someday goes away, what are they going to do with all those samples? And again, the answers were all over the place, and this is something that will probably follow up in the next round. But most of the answers were saying that a virtual biorepository system or a directory in a catalog of where these samples are would be something very useful, so that they don't disappear, that people will have access to them for research and other uses. So getting to the barriers to specimen sharing, which May has introduced before. And of course, it was in great detail discussed by Nikos about what some of those barriers are in shipping and sharing specimens. In order to be able to share specimens, people have to be able to ship them. And for any of you who during the pandemic tried to ship specimens, I think you'll feel this very acutely. There were no flights in, flights out. Things were getting hung up in customs and all kinds of headaches. So shipping is one of the major hurdles that people have identified in the process of being able to share specimens. The question here was, again, what was people would rank as the highest, most greatest barriers they have in sharing specimens in the process. The restrictions that have been described earlier would be the highest there. Issues of cold chain and just ability to ship the specimens. The regulatory part was also, and then finding sources of specimens was also ranked. But the highest were kind of the restrictions that have been earlier discussed. These were some of the other barriers that people have talked about, and these are questions of infrastructure, again, thinking about where the specimens are and where you have to get them from, often questions of just having the basic infrastructure, backup systems, and so on. Funding is not a trivial one, because you need the supplies, you need staff. For shipping, the cost of shipping is not trivial. The regulations we have heard about. Lack of knowledge of those regulations is also a barrier. Confusion about what is or isn't applicable. In the US, of course, importing things in the US, I think, as Nikos explained, can be a challenge. And for that reason, the way FINE is doing, maybe taking the test to the sites rather than trying to get the samples in would be one way of doing. Negotiations around material transfer agreements is a barrier. Some organizations overvalue the samples themselves, so that makes it difficult to kind of negotiate and to do this in a timely manner. And then sometimes just simply access of how to get the samples from areas of conflict areas and where there could be outbreaks. The ethics is not a trivial issue. The consent that has been discussed a little bit earlier. And then a very important one always is really the data. Access to the data and having complete data and complete annotation of the specimens. So again, these were the questions that were asked. What are the type of benefits people would see from having a virtual biorepository system that would help with the logistics of the user and the source of the specimens getting this done. Having templates for material transfer agreement was one that ranked highly. As some of you may be familiar that the World Health Organization's BioHub have trying to put together some kind of generic templates to help people negotiate, and one for commercial and for non-commercial entities. What would the other benefit, great benefit of being part of this virtual biorepository system would be opportunity for networking and collaborations. Capacity building was ranked high. Opportunities for training, such as GCLP, for example, would be important. And then help with basic biorepository functions, just this idea of coordinating the logistics. And then expertise, access for people who don't have a lot of expertise with biorepository. That's something the virtual biorepository system can do. And then providing standards and SOPs would be another. These are some of the services that the virtual biorepository, the question was, what are the services that people would consider important for this virtual biorepository system to do? And again, these are the very high kind of things that were ranked as most important. Reference panels were ranked very high. The infrastructure and the logistics help. Protocols with SOPs and templates were also, all of these are of course important. The training and then the assistance with the negotiations. One of the always big issues around biorepositories is how to keep them sustainable. So with the virtual biorepository system, because it is not a huge facility that you are maintaining, the samples are distributed. The cost is not in the virtual, in the maintaining the samples because they are maintained locally, but still, how would you be able to keep this as a sustainable system? And everybody, it was pretty much 100% consensus on all the answers that some kind of cost recovery would be helpful. And they all agreed also that there's some type of sliding scale on fees for services would be helpful. And there wasn't necessarily agreement among everyone on how you would make that happen. But I think that there was consensus also then at in some way making it more easier for people from LMICs to be part of the virtual biorepository system and also use its services would be make it more equitable. And another possibility would be that, for example, if there is some fee for service for a commercial entity, then whatever that's collected, and there is a contributor who needs a freezer or whatever, this would be, so you have sort of like a frequent flyer kind of system where based on contributions, you could get access to some help that is coming from these cost fees for services. Outside of LMICs, people were divided about how that would work. Some also agreed to maybe a sliding scale and suggest sort of complicated algorithm that you would, based on what service you are providing and what kind of funding you have and so on. And I'm not sure if the respondents thought out clearly on who exactly would administer that, but that was one suggestion. But again, the consensus is trying to build something equitable. So to summarize, again, this is our end of round one of this Delphi survey, and we're going to go into the next round. But what we got out of this, a little bit about what type of specimens people would be interested in for immunological type applications, blood and derivatives collected at various times, specimens from endemic disease areas, the utility of collecting some high volume specimens for specific needs, setting criteria for inclusion, quality criteria, and maybe a tier system for different uses, and then having a minimal required data set for making these specimens useful. to overcome some of the barriers that have been identified, shipping and ability to share between countries. And I think Nikos has gone into quite a bit of detail of what some of those are. And the alternative maybe testing in country rather than trying to collect the samples. The ethical use and consent issues. So in the US, there is a possibility of broad consent. I think there is something similar in Europe, but it is not universal. In many places, you cannot share specimens for unspecified future research. You have to have. But I think that the respondent said that if it were clear that this is for type of immunological kind of applications, and then that could be worked out as maybe amendments to the consent or to their protocols and included in their consent, of course. And so what type of benefits and services I just mentioned already. The reference panels for proficiency testing and calibration were very highly ranked templates for protocols, templates and protocols, material transfer agreements, and then standard operating procedures and so on. The logistics support of making it happen, go from one place to the other. And then other benefits that were mentioned in this survey, networking and collaborations and training. This is something that we have heard from previous survey workshops that we have done, that people would consider that as a benefit to belong to this virtual biorepository network. From this round one of the Delphi, there's high level of interest in biorepositories and the issues of access to specimens. And many people have volunteered that they would be willing to participate in developing sort of the governance for this virtual biorepository system, the policies for access and use. And appeared again that there is greater interest in this idea of a distributed federated system and compared to centralized biorepository. Again, with the caveat that, that it depends what the biorepository's purpose is, but for the purpose we define, that seemed to be the consensus. So this is to summarize then that, this is what the Delphi kind of consensus survey process has shown us, that there is a high level of interest for a type of virtual biorepository would be supportive of other concurrent efforts and would facilitate equitable access to specimens, services, and benefits. And that there is room for clarification and perhaps in the future for more the type of talks that Nikos has given about what are the regulatory challenges, what are the ethical issues. We haven't gone into detail about that, what those would be. So that would be helpful in going forward to provide some additional information on that. So to finish here, we are extending an invitation here to consider joining this pilot that we are trying. I mentioned we want to recruit about 10 contributing members to the virtual biorepository and start out and would like to have those who participated in round one who are interested in the Delphi to continue to participate. And then we want you to become all part of this network of the coalition of the willing, as May likes to call, to solve the current barriers and to provide equitable access and timely access to specimens. So thank you for your attention. And for any of you who are here who participated in this survey, thank you for that.
So we're just gonna skip over that, any questions to later. If you want our comments, please, because we're running a little short on time. So this will allow us to move forward. So I think you heard then what this, was it 236 that we reached out to? They're actually down they're selected from a larger pool of over about 700 persons that had indicated interest in biorepository ethics and use from previous workshops. And so these are really biorepository, it's biased towards biorepository activity supporters, right? And we're asking them then what to do next. So this is pretty much not just from the street, but really people who know something about it and what to do, and they're advising us the next steps. So one of the things that maybe we have the next set of slides, I think there's only 1 or 2 slides. So what are we talking about for our biorepository? And I think also that we'd love to have discussion with Warren and later, is that we're really talking about, the founding contributors to this the conceptual virtual biorepository we like to launch from here in Cape Town this time around. And that is, what should be considered for these. And that I think what we need to do is make sure that we deal with the ethics, that we look at the type of serological samples. And the serologic samples is focused on because there are larger exchanges of samples as FIND is doing. There's also the WHO BioHub that's exchanging viruses. There's also another activity called the European Viral Archive Global that are also providing access to virus isolates and strains. But there's really no big place that does the characterized immunological serology samples that we're talking about. And this is the reason why we feel it's an entry point for what we can do and show. And then later on, we want to make sure that it's in step developed with the other activities. We're talking about 10 mLs, that a site has to be able to contribute up to 10 mLs of 10 different samples. That's still generic at this point. It's not marked at this time because we want to have further discussions with everyone on what's next to do. And that we want to define the role of the founding contributors. And mainly it's really because the role of the founding contributors is to help us sort out some of these grassroots items and issues and questions that we have. Because rather than having the policies and all the rules now and the ethics now, what we want to do is work from the grassroots with the 10 volunteer founding members to be able to articulate and define and adapt from the others what is really important for us to make us successful. Okay, and so those are the activities. I think that's it. So, okay, so I think we're good. And I think our timing is just perfect because we have Trudy Lang, who just joined us, and she's here to say a few words. Most of you know her. She is the director of the Global Health Network, a key partner and person that I've loved working with over the years. And we keep saying that there's someday we will work together doing something, right? And here we are. And the organizer of this conference, the person who's helped us with her wonderful team, help us before with the workshops and now preparing for this pre-conference workshop. And then we'll be having a booth at the for the next 2 days to really have you stop by and talk to us about many of the things. But I think I'll just welcome Trudy. It's lovely to see you. I haven't seen you in person. Okay, please.
Thank you. Oh, thank you. And it's this is just such a nice, important day, actually, to be able to have these small workshops and symposia before we get underway with the conference tomorrow. And biorepositories is just such a perfect topic to cover. So thanks all for coming together today. And this is such an important focus to have. And it's just really great because I've got this crazy schedule that they've given me, and I'm popping from symposia to symposia, and it's just so great to see the focus on the topic. So I've gone this morning from the networks of excellence across Africa, where we had colleagues even coming in from Bangladesh and Nepal and Brazil. I've just been at the AMR symposia, and then here you are for biorepositories, which is cuts across everything, right? And it's so fundamental. And it's one of the most important topics. And how many years ago did we first meet? We said, we have to do this. We have to do this. So it's, I really want to just thank you and then just say, right, how do we properly do this now? And it's just such an opportunity we have. So if you don't know much about the Global Health Network, I'm apologizing now for we're going to bore you rigid with that over the next few days, because it's going to be the sort of focus of what we're talking about how we've built that over the last 10 years or so, and how the whole point is to be a mechanism for connecting up groups on these really important cross-cutting topics. And, you know, just like AMR, which is so related to biorepositories, of course, because we need sample repositories and we need data repositories, and we need to have the ability to connect between disease areas, between geographies, between teams, and work out how we can share that best know-how. And so I think for the opportunities we have to really transform biorepositories, we can use what we've learned around this whole philosophy and action around communities of practice, which is effectively what the Global Health Network is. It's a digital platform. And yes, we've been very fortunate. We spent 10, 15 years developing the digital platform, which enables that transfer of knowledge, like osmosis from areas of high skills to low, and making that movement of information between diseases and teams and groups. And we've invested probably 6 or 7 million in the digital tech that can make that happen. But what we've also done over the years, and it's been just great, just walking around today and meeting some people in person I've either not seen for 10 years or only on a screen. There's now a really strong footprint in the regions as well. And you'll hear over the next couple of days about the Global Health Network, Africa, Asia, and Latin America. And we've been funded, even, initially, we've got 47 coordinators who are going to be capacity development people, or they're working on data science, or they're working on a particular topic, maybe the research nurses. And I think when we look at how we can sort of build on that and utilize that opportunity for biorepositories, it'd be great to think about some regional coordinators. And in the starting point, we can leverage the teams that are there already, and we can run workshops in these places where there are already these people in place, and they can run biorepository workshops or a clinic or whatever it might be using. You'll hear all about this opportunities the next few days. And then over time, as funding comes in, you could have biorepository coordinators that can use that system and that infrastructure. And it's like a franchise operation, really. And I think that's a really unique and important opportunity to leverage for really taking this to scale. I think the other thing I want to say about biorepositories, so I've been running around trying to support, better clinical studies and practices for almost 30 years now, and most of that has been in Africa, but a lot in Asia and Latin America too. And I spent a long time in Kenya at the Kenya Welcome Program, and they always are very proud of their biorepository of 35 years more of amazing malaria samples. But I was in Bangladesh a few weeks ago and met this incredible team working really with not much support and not really discussing with others, and they've got this incredible climate change potential biorepository because they just happen and haven't really done it in a formal way. They've got water samples and they've got clinical samples, and how salination has changed in this one area, and they're interested in what the outcomes are on pregnant women, or aging, or diabetes, on the different mineral content in the water. And he said, this is just a perfect example, and I was thinking I need to talk to Matthew about this. They have all these water samples, soil samples, blood samples. And then what do we do with these? Well, exactly. And he's got, 25 years of cumulative samples from that story. So it's everywhere, and it's so ubiquitous. And this is the point, isn't it, about how do we transfer know-how from the groups who really have got these really state-of-the-art biorepositories and mechanisms for sharing, and then others who want to, or even setting out at the beginning. And I think everything that the Global Health Network is trying to do about that transfer of information between disease areas, regions, teams, and goals, that's precisely what we need to put into action for this. And I think the other really key point is around this point of trust and the philosophy of community of practice, because this is around, and I think you can almost talk about biological sample storing and use as you can data sharing, sampling. and data use. And whenever I give talks to data groups now, I keep saying, can we stop saying data sharing? Because I think it's just upsetting everybody and it's becoming meaningless, because largely data doesn't need to move anymore. Data can stay where it is, and you can answer the question of the data in situ. And if that can largely happen as well, biological sampling as well now, right? You can have samples, those samples don't need to leave to Bangladesh, but we could work with the team to help them analyze whatever questions they want to ask. And I think the perceptions of the old days, and we're going to talk about this over the coming days, where we've had, the old models of capacity building, which was really to teach people to collect data that's taken off elsewhere. And it's the same with samples. I first came to Cape Town years and years ago because we were doing PK samples on malaria drugs. And it was just, sweeping up all these PK samples from across Africa and taking them to a lab in Belgium. Well, that's not how and anything like needs to happen anymore. And so, it's around access, isn't it? And asking questions of existing samples, increasing laboratory capacity, or connecting up in a region even. There might be a really good lab working on malaria in one hospital, and then somebody's got some samples working on leishmaniasis or something, but you can share that tech and know-how even within a hospital, and that often doesn't even happen. So that's the point. I think we need to just get smarter around some of the language we use and changing the perceptions. That's I think when people say sample sharing or repositories, they expect us to be saying, I'm asking you to give something away, which is not what we're doing. And I think there's such a big piece of work to do around that. So the last thing I wanted to say really was the opportunity we have today with this group, and then going forward over the next couple of days at the conference. I don't know if you've got your conference bag yet, and you can laugh. Made sure to tell me. I wrote a letter to everybody saying, okay, this is a really important opportunity. Did you read it? Was it okay? It wasn't too rude. Zaina would be laughing. But so in the conference letter, I said, every lunchtime, every coffee break, don't sit next to somebody you know. And there's no parallel sessions apart from today for the symposia, but it's the next 2 days. It's all one session. So, sit with somebody you don't know. And it's all about recommendations. It's deliberately cross-cutting, which is perfect for biorepositories. And it's about bringing different, types of research from different organizations together. And we've also got a really important opportunity, which I don't know if Mechu's mentioned yet or you've had communicated, but PLOS Global Public Health, we're going to run a special collection on the conference. And so that's a really nice opportunity within amongst all of that outputs is I think like our, whatever you want to call it, recommendations, mission, declaration, whatever you want to say, right? Biorepositories, this is the really important outcomes from this. So, where do we take this next? What can we do? There's so many people, and in the conference book, and there's a delegate list, you can, hunt people down, pin them in the corner with their cup of coffee and say, do you have any biorepositories in your, what do you do for, are you interested? Do you want to take part? Build that community and contacts and build a plan as well. And I think the opportunities for that PLOS paper are really exciting, and it could take any form you want. It could be a kind of review of where the world is. It could be where you want to take this. It could be the opportunities. It could be a call for action. It could be a mixture of all of those things. But I think a PLOS paper from this would be a really nice capturing. But I think whatever you decide to do with it, it's about getting clear recommendations that you can take forward over the next 2 days, but then from there on. So thank you, Mechu. Just the nicest thing to see you here, you and I together in Africa. Perfect. Thanks everyone for taking part. And I really hope you make the most of the next today and the next 2 days. And good luck. Thank you very much.
Okay, so I've been tasked with giving a sort of overview of biorepositories, or a Biorepository 101 course. And I'm going to sort of march through it by talking about the kind of people that are involved in biorepositories. I'll talk about considerations for setting up a biorepository, some various biorepository models, and I'll get into the regulatory and ethics stuff at the end. And if I don't get through it all, happy to talk about it with anyone as we go along. So the reality is, I think biorepository directors sort of go through 3 phases. Most of us start out as researchers who begin their biorepository, but it's not really a planned activity. It's something that you're just doing. As Judith and I were talking about at the break, you're really a collector. You're not a biobanker per se. And this is what I call the accidental biobanker, right? Someone starts out a collection as an outgrowth of their regular research. They're hanging on to samples just in case, right? I may want to be able to do something with this later, but it's not really a planned activity. Record keeping is often similar to other things that you do in the lab. So if you're one of those people who's really, document focused, you'll have a really good record. A lot of people, it may be a paper record that's of varying degrees of quality. Some people may simply have their inventory in their head and not actually have any kind of true inventory. Yeah. And there's no judgment in what I'm saying here. This is just a statement of fact. But often the people who have these collections really haven't thought through all of the regulatory issues around second use. And some of this discussion came up earlier today, and it's a really important discussion. Then people move on, and some researchers will get involved in larger projects, things like clinical trials, where biobanking has to be a bit more formalized. Usually there's a procedure involved, you'll have training, there's documentation, there's probably an inventory system. And this is what I refer to as the incidental biobanker, right? So someone storing samples temporarily for a larger study, you have to be organized, but you're probably not keeping them all. You may actually be collecting samples for personal use as a part of that larger study. You may have a sub-study that you've gotten ethics approval for, but it is, you're doing it in an organized way, but maybe not to the level of a full biobank. Record keeping at that point is often going to be dictated by the study. So you may have a web-based system or some other system that you're logging into. Whether or not you'll be able to use that same system for your own collection is always a question. You may or may not have permission to do that. And when you leave the study, you may lose access to that. So it is a consideration. But sample labeling becomes standardized. A lot of the procedures become standardized at that point. And so you move into a realm where you feel like, oh, I may be able to take the next step and actually do a full biobank. And then a small number of researchers will actually then realize they can do it at scale. They can actually start to compete for grants that require biobanking. And there are a lot of grants and contracts out there where this biobanking facility allows you to be competitive. And so you become what I call the intentional biobanker, right? You have organized storage, organized record keeping. You move toward electronic records, if at all possible. You have equipment that's monitored and recorded so that you can talk about the value of the specimens that are there. And you may end up moving towards various kinds of certifications, right? So ISO or GCLP, things that show that you are at a particular level and that when you apply for funding says, yes, I have the ability to do X, Y, and Z, or X, Y, and Z since I'm in South Africa. Okay, so setting up a biorepository, there are a lot of considerations for when you set up a biorepository, and a lot of people don't think through all of the details about this when they're starting out. The truth is, very few institutions want to pay for biorepository efforts. Biorepositories are expensive. They are not cheap at all. Biorepositories take up space. And I don't really care what kind of institution you're in, space equals money, right? Administrators will be thinking about the space that you're occupying and could it be better used for some other activity. So, it isn't always about shillings or rand or dollars, but it often is. But more importantly, it's thinking about what your institution values. And do they see this as a valuable exercise? And if so, they may invest in it. If they don't see it as a valuable exercise, you're going to be fighting for all of these resources as you go along. And the reality is, when you're talking about expanding, buying freezers, buying liquid nitrogen tanks, electronic record systems, all of these things, sometimes you can put those costs onto your funding sources and sometimes you cannot. So one of the biorepositories activities that I have is for an influenza program. And I had to agree to take on 400,000 specimens as part of that program. And I was strictly forbidden from buying any permanent equipment on that program. I had to find a way to fund it through my institution. Now, indirect costs allowed me, I was able to make an argument and get a carve out of the indirect costs to be able to do that. But I had to know that up front, and I had to know what the costs were going in so that I could actually make that happen. So when you're thinking about setting up a biorepository, there are a lot of considerations. What's your physical footprint? Do you have the space? Are you going to need more space? How are you going to grow? What's your infrastructure? What's your backup power? Are you in a location where power is lost frequently? In which case, not only is it challenging to keep the freezers running, but every power up, power down cycle creates stress on those freezers and makes it much more likely that they're going to break down. What's the physical access to the space? And there's two aspects to that, right? How easy is it for you to get in and actually access the repository or move liquid nitrogen dewars in or out, right? If you're on the 10th floor of a building, how hard is it to get things up and down? But also, do you have the space locked down? Do you have security? Can any random person walk in and potentially make a challenge to your space? And by the way, I'm more than happy to share all these slides with everybody. So if you feel free to take pictures, just saying I'm happy to share the slides. You have to think about the record keeping, right? Paper records are great, but at some point you probably have to convert over to an electronic system. And when you make that change, it will be painful. I can tell you, it will hurt. So I'll give you an example. Like my wife and I collect music and books. You know, my wife is a huge reader. I'm a reader, but not as much as she is. And I'm a big music fiend. And like, if you walked into my house and said, do you have a Bible, a copy of Stephen King's The Stand, or any Dorothy Comson books? I could say, yeah, I have all of those. And if you asked me where they are, I'd say, let me get back to you on that. Where if you asked me, do you have Beatles albums and Fela Kuti and, you know, Dave Brubeck, I could say, yeah, and I know exactly where they are because at the beginning of pandemic, I actually inventoried all that stuff and I know exactly where it is. But, you know, doing that kind of transition is challenging and you have to invest in it. It is something that requires time and effort. And to be able to participate in something like the virtual biobanking effort that was described earlier by Warren, you kind of have to have that stuff in place. And so when do you make that transition and whose effort goes into it, right? And how do you pay for that? And then, of course, there's all this stuff about metrics of quality. Like, are you monitoring the freezers? Do you have good records on that? Are you sure that the samples that you're storing at -80 are really at -80? Or if they're at liquid nitrogen, are they really at the right temperature? You know, can you document that so that you can show the people who are going to use the specimens what the quality is? But there are lots of other considerations, right? Do you have a labeling system? What labeling system are you going to use? Is it going to be 1D barcodes, 2D barcodes? How much information are you going to put on the vial? Are you going to put dates on the vial? Because for us in the United States, a sample with a date on it, that's protected health information. That changes our regulatory landscape if we have a date on the vial. What's the sample metadata? And who holds on to that metadata? Because the reality is most people who are biobankers don't hold the metadata. The metadata is in a separate database. You can link them, but how do you do that? And what's the regulatory landscape for doing it? What's your staffing, right? Do you have dedicated staff that you're paying them? This is the only thing that they're doing? Or are you taking your lab staff who obviously have assays to run and data analyze and all of that kind of stuff? And oh my God, samples have arrived, drop all of that and go do this. And there's no right answer to this. I've actually done it both ways, but it is a consideration because you have to pay people. People don't do this work for free. So there are financial considerations when you're doing all of this. What about training? Do you actually have a training program? How do you go through all of that? Do you do proficiency testing? Do you show that the person you've trained actually can do what they're supposed to be doing? And how do you prove it? And then do you have external evaluators come in and see whether or not what you're doing meets a certain level of standard? And all of these considerations come in, and you don't have to necessarily go to the most extreme level on day one, but you should think through, where do I want to be, right? How far down this pathway do I want to go? Because the further down that pathway you go, the more competitive you are for other funding opportunities. And so quality oversight becomes a really big, important factor in all of this. I have external evaluators that come and make sure that we're doing what we say we do. And when we sign on for a new company study, they come in and repeat the same thing. And we're at a point now where we're like, yeah, come on, fine, inspect us, we're good. And if they find something great, we can learn. That's awesome. You need to think about chain of custody, right? How do you prove that the sample that was drawn from the patient is the same sample that wound up in the freezer? Can you document that it was handed from this person to this person to this person to this person? And that's actually a really important thing, especially when you're talking about the kinds of samples May was referring to earlier. You know, if you want to be able to say this is well characterized, that's part of well characterized. How do you reconcile errors? Mistakes happen. You know, errors are going to occur. Do you have a process for dealing with that? I mean, that's really important at Reciba because there's teeth behind all of the laws that you're dealing with, but it's true for all of us, you know, that you have to have a procedure for dealing with it. And the procedure is not, hide the error. No. Just document everything. You need to have procedures for discarding and destroying samples and documenting that you did it. And finally, deviations. You need to have a process for deviations. A lot of people view deviations as you've screwed up and nobody should ever have deviations. I take the opposite view. If I see a laboratory that has no deviations, then they're either lying or they don't do any work. Because we are humans and mistakes happen. The best thing you can do is go, yep, something happened. Here's the documentation. Here's what we learned from it. Here's how we're going to try to not make that mistake again. We'll make all new mistakes in future, but we'll try not to make that mistake again. So when you're going to set up a biorepository, I think there's a bunch of questions you should ask. Like, number one, why do you want to run a biobank? It's a pain, seriously. Like, there's a lot of headaches in it. But if you want to do it, you should go into it consciously. You should make a positive decision about it. Importantly, do I understand the regulatory landscape? And perhaps even more importantly, do I have the staff necessary to help me navigate it? Because the regulatory landscape is there to protect the individuals who gave the specimens, or to protect the wildlife and the environment that you're in, or they're there to prevent the release of something that shouldn't be released to the larger world. And don't view regulations as an impediment, view them as a way to get things done, but it does require staff that can actually help you do that. Because the first answer is almost always going to be, no, you can't do this. What you need to do is you need to have people that go, we can't do it right this second. Here's the pathway forward. Here's all the documentation that we need to cover. And they need to stay on top of the regulations because the regulations change over time. It isn't that what I do today is the same thing I'm going to do 20 years from now. Heck, the thing I do today may not be the thing I do next week because the regulations may change. You need to think about disaster planning. What do I do in the case of a flood, a fire, any of those kinds of terrible accidents that can occur? You know, do you live in a floodplain? Do you live in a place where hurricanes come through and the biorepository needs to be on the 6th floor of the building so that if an 18-foot storm surge comes through, that you're not flooded, right? These are real considerations. And there have been some pretty impressive disasters that have occurred with big biobanks where they didn't have the infrastructure necessary to deal with it. Now, of course, you know, an earthquake in a place that's not earthquake prone, there's some things you just can't plan for, but you should think through, like, what are the common problems we have here and how are we going to prevent them from creating an issue for our biobank? What do you do in times of surge? And I don't mean storm surge here. I mean, I have 3 studies and all of the samples are arriving on the same day. You know, do I have enough people to actually deal with it? Are there people I can pull in to help do all of the processing so that the processing happens in a reasonable period of time? We went through a lot of this with COVID, where suddenly we went from processing, you know, maybe 10 specimens a day to processing 70 specimens a day. And we had to work in shifts. We had to bring in people. We had to do some pre-discussed deviations to say, yeah, we would love to have people who were already trained on this. We know they do good work. We will watch over them, but we just don't have time to train them up at this point, you know, that kind of thing. And again, you know, will taking staff for the biorepository stop your regular lab work, right? The biorepository is rarely the only thing you're doing. And is it going to mess up the other part of your portfolio? If biobanking is the only thing you do, great, then you can fight with the administration for people. But, you know, think about how it's going to impact all of the other parts of your life. Do you have enough institutional support to carry yourself over if funding wanes, right? Our institutions love us when we bring in money, and they're less enthusiastic about us when we're costing them money. And that's just a reality because they've got to keep lights on and doors open and labs running, you know, and I don't want to make it all about money, but frequently the administrators are having to think about money whether that is money coming from the government or external funding agencies, they do have to think about how are things going to get paid for. Are there other biorepositories at the institution? This is a big deal. At Duke University, there are about 8 reasonably large biorepositories. I am one of them. I probably hold more specimens than most of the others put together, but I am not the favored biorepository at Duke, right? I'm not the one that gets the dean's subsidy. I'm also the one that doesn't ask the dean for any money. So I have a reasonably privileged position, but am I going to have a time where they may say, well, you should shut down in favor of these other people? And I can make an argument and say, but see all the money I'm bringing in. Do you really want to do this? But think about, are you starting something that already exists, or are you creating a new thing in your institution? Is there institutional funding for the biobanking, and is it secure, or does it depend on the person sitting in the president's seat or the dean's seat or the chair's seat or the division head seat or whatever? Or is it a broad institutional initiative, right? Because if it's tied to a person and that person leaves, gets fired, gets promoted, whatever, is that going to disappear? And it doesn't mean don't do it. It does mean think ahead. Think about where all of this is going. What do you do if the funding for a collection runs out, right? This is a really common problem. Study going on, you've collected all these samples, suddenly the person who was doing all of that, they don't have any funding anymore. What do you do with those samples? Do you hold on to them? Do you get rid of them? Do you give them to somebody else? How are you going to deal with that? And there's no single right answer to that, but you should think through those problems up front. And importantly, what do you do if a client can't pay, right? You've done all this work, and then suddenly you're out all of these resources that they're not going to recoup you for. What do you do in that situation? How far do you get into it before you say, I'm sorry, I can't do anything for you anymore? And we've all run into this where people are a little more overeager in their planning. Related to all of that, I personally think you should think about a biobank as a business. Now, most of us went into science because we didn't want to go into business. But the truth is, you need to know what your costs are, right? You need to know how much it costs you to do each of these activities. So do you know how to cost things out? Do you know how to budget, to create a budget? I am always amazed at the number of scientists that are terrible at making budgets, which always blows my mind. I'm like, you can sit down and write out an experimental protocol. A budget is just an experimental protocol, but instead of milliliters, it's money, right? That's really all it is. But can you do that? Do you have the support to do that? I think you should make cost estimators and review them annually. And what do I mean by a cost estimator? I think you need to learn what it costs to do a specific activity. So, for example, I have a budget in my lab for what it costs to do an ELISA, right? I have a budget in my lab for what it costs to isolate serum, for what it costs to isolate PBMCs. And what I did is I had someone do the experiment and literally count everything that was used, every pipette tip, every serological pipette, every pair of gloves, every bit of PPE. And we made a list. Here's everything that was necessary to do this activity. And this is what it cost me to buy all of those things. And so here's what the marginal cost is. And so at the end of the day, this is how much I have to spend to do the activity you want me to do. So if you want me to do that activity, here's what it'll cost you to do it. Once you have those cost estimators, applying for grants and contracts becomes so much easier, because then you can say, oh, I want to process 75 specimens at these many time points, 75 people, this many time points, these are the specimens I'm going to collect. It is literally then just my cost estimator times what all of those numbers are. Boom, here's the number I need, right? Once you have that in hand, it makes life so much easier. Somebody told me this early in my career and I bought into it, and it has made applying for grants so much easier. Okay, let's see here. Yeah. Ask yourself about storage. How much does it cost you to store the specimens, right? Is the electricity being paid for by you or by your institution? Do they charge you for that electricity? Do they charge you for the space? Do they charge you for liquid nitrogen? Like I keep a lot of samples in liquid nitrogen, and until I moved into my new facility, I was paying $40,000 a year, right? So roughly 700,000 Rand a year just to move tanks, not even for the liquid nitrogen, just for somebody to take a tank, put it on a truck, fill it up, bring it back, and give me the tank again. I was paying a ridiculous amount of money for that, right? And you have to figure out how to budget for all of that. You have to ask yourself, am I going to charge for the storage, or am I going to wrap those storage charges up in the processing fees? There's no right answer to that, but you should think through it. And do I charge for shipping, right? We were talking about this in terms of the fine biorepository, and what Nikos was talking about. Shipping is not cheap, right? And it can get really complicated across international lines. Think about, are you going to focus on a specific core activity? I'm going to process sera, right? And that's it. Or are you going to offer a diversity of things that you'll do, which then will attract more customers to you, right? And so we made a decision early on that we wanted to be more of a full service shop. So we do processing of tissue samples. We do oncology studies. So we get tumors and we take pieces and freeze them away and separate cells and freeze them away. We have rates for all of these things, and we can then have people pay us to do lots of other things, which allows me to have a bigger staff, which actually makes it easier to buffer the highs and lows. But it's taken me years to build this staff. And so you just need to think of how far do you want to go in terms of building a biorepository, and what are you going to do with it? Do you ever tell somebody no? And the answer is yes, you need to be able to tell people no. I have had a number of investigators who came to us, we did a study with them, and they were so difficult to deal with that when they asked us to do another study, we said, thank you, but I think you should talk to these other 8 biorepositories on campus because, thank you. We just don't have the bandwidth, right? I'm sorry, when in reality it was, I wouldn't help you if you were on fire. Like, it was that level, like, seriously. And then can you balance being helpful, but also being respected, right? You want to be seen as a valuable resource, and you don't want to be a doormat. That's a key thing. So yes, you want to be helpful to people, but people also have to respect your boundaries, right? So one of the challenges we've had is, we want to do all of this specimen processing. We're going to collect those specimens at like 9 o'clock on Saturday night. Somebody will be there to process it, right? Kidding me? Like, really? I've got to now figure out how to staff it. And then, Sunday comes along and they're like, no, it didn't happen. I'm sorry. We should have called you. Yeah, I'm sorry. So you've just paid people to be on site and wasted their time, your money. And, so you have to set boundaries is what I'm trying to say. You have to actually set boundaries. Okay. Now, biorepository models, and really what I'm talking about here is thinking of how do we discuss the ways that you can set up these virtual biorepository or larger biorepositories scenarios. We all know that samples have value when they're used, right? And unfortunately, samples are really not like wine. They don't acquire value as they get older, except for that very small number that do, right? There's a handful of things that will become more valuable. Being able to pick which of those samples will be more valuable is really hard because we don't have the ability to predict the future. So you've got to think about what are you storing long-term and what is the value going to be? But the truth is we all worked really hard to get those specimens. So we don't want to see them wasted, right? You don't want to just give them out. You want to make sure that whoever's going to use them is actually going to use them for good. And we also want our hard work to be recognized, right? You don't want to just be, selling the samples off to somebody because that's not really appropriate. You want to make sure that your work's recognized, that the patients are recognized, or the work of the people collecting the animal samples are recognized. And, we have to come up with a way to do that in a virtual biobanking system. And I think, the stuff that was talked about earlier by Warren is a good way to do it. Let's get users and sample holders to talk to one another. But there's several different ways one can do this. Now, the reality is nobody wants to be the person who gives away the sample that leads to, a Science, Nature, or Cell paper, or goes on to win somebody a Nobel Prize, right? But I could have done that. Yeah, the fact is you probably weren't going to do that. But you also want to make sure that if that paper is written, that you're acknowledged in there somewhere, right? That's important. And nobody wants to be the person who sells a high value item for less than it's worth. So in the US and actually in the UK, there's this show called Antiques Roadshow. I don't know if there's something similar here, but it's the idea of I went to a yard sale or an estate sale and I bought something for nothing and I take it to an appraiser and they're like, this is worth a crazy amount of money. You don't want to be the person who sold that for nothing, right? Nobody wants to feel like they've been taken advantage of. And so how do you protect yourself against that? That's really the question. We all want to believe that our colleagues won't take advantage of us. And in general, I would say that's true. We all have this fantasy that this is what science looks like, right? We're all standing around and we're collaborating with one another and, we're all working together. And the truth is, when you start collecting biospecimens, you tend to look more like this, right? I'm Smaug sitting on my treasure hoard and this is mine. And, I'm going to fight you for it. That's the way a lot of us tend to be. So I'm going to present 5 different models for specimen collection and sharing. 2 of them are distributed and 3 of them are centralized. And you could hybridize or combine these things in any way you want. And I don't want to make it sound like this is all original. This is just me collating ideas over the years. We've actually talked a lot about this. These ideas have been in the community for a very long time. And they're just conceptual frameworks, really, for talking about biorepositories. And all of them assume that the people collecting samples of interest that they're collecting samples of interest to them and potentially to collaborators, right? And it's just really meant to be a starting point for us to talk. So the first thing is the private collector model. This is actually where we all start out. That is, you're collecting samples for yourself, and you're storing those samples on a limited basis. You might be willing to have somebody broker a deal where something might be able to go out, but the reality is, you're hiding it all. You're keeping it all for yourself. I worked really hard to get this. This is mine. And it's the most restrictive of the models. Oops. The museum model is really a lot of what we're talking about here. And it's actually, or a hybrid museum model is actually exactly what Warren was talking about earlier. You have a collection and you might be willing to share a catalog of that, right? Nobody can walk in and take a painting off the wall, but they could see that, oh, you have that painting. That's lovely, right? And you could actually have a coordinating center that could act as a collector of catalogs. And then people could look at those catalogs and say, let's negotiate access to those things. And that is a kind of virtual biorepository model where you could actually hybridize it and have the status be, at a limited view. For example, if you have select agents, do you really want everybody to know that your select agents are located at, this specific address at this specific location, right? I mean, I've seen those movies that they don't end well. So, you could actually restrict access to that information. And there's different ways that one could do that. There's the idea of a bank model, right? So that you could actually deposit your material into a location like a safe deposit box, but you retain control, right? You're actually they're holding on to it, but it's really your item. And you're only going to be the one who can get permission for it to leave. The bank retains the physical item, but you retain the control. And again, you would have to negotiate access. There's a bookstore model, right? Where basically you're saying, yeah, I have these samples. I am willing to pre-specify some conditions. I'm going to give you these books. Here's the price you need to sell them at. It's essentially what it is. You would retain the connections, right? And you would actually have the ability to say, if they meet this price, good, blessings be upon them. If they don't meet that price, well, maybe we should talk. And, maybe we could negotiate for a different price. And that's a more centralized kind of model, and one that I think a lot of people favor. There are challenges with that, right? Because you're ceding a portion of that control. And from an ethics perspective, it can get really tricky. And then this is the one I think a lot of people think of when they think of a centralized biobank. It's the thrift shop, right? You've given over your materials, right? The thrift store sets the price, but there's no real connection back to the originator. And it does give the most rapid access, but it probably also gives the least back to the individual who collected the specimens. And importantly, you lose a lot of that information, right? Because ethically you can't share it. So, the truth is the specimens are really important, but we have to ask, like, how much of the information needs to go with the specimen, right? There are critical pieces of information we need to know, but if we strip away some of that information to make it easier from an ethics perspective, is it still valuable, right? And how much of that needs to be there to retain that value? Is it simply year of collection? Is it simply male, female? Is it simply age and years? You know, how much of that information needs to go along with the specimen, and what is permitted to go along with that specimen? And then, of course, you know, and we've talked about these multiple times, you know, cui bono, who benefits? And, you know, there's obviously all of the things we've put in place to try to protect our homes and institutions. Nagoya intellectual property rules, you know, we all need to get promoted at some point, right? And so, you know, how do you get the recognition that is necessary so that you're deriving benefit from these activities that you're doing? But then there's this, which is really important, and that the second Latin quote there is essentially who watches the watchers, right? So who is paying attention to the person making the decisions about the distribution of these samples? And this is where the ethics part really, really comes into play. It's the most important thing. And, you know, we've talked about this quite a bit, you know, as we've been discussing ethics here. I mean, the reality is I possess the samples. I do not necessarily have control, right? So, and this is a really important concept for biorepositories. Possession does not equal permission. Just because it's in my freezer doesn't mean I have the right to give it away or do anything with it, really. I have to figure out the pathway forward from a regulatory perspective. The way that I like to think about this is in terms of a legal trust, right? A trust in the legal sense really defines three different parties. There's the person who grants whatever it is being held in trust, right? If it's money or if it's the specimen, you can think of this as the person. There is the protector. What body is charged with making sure that this is being used properly? And then there's the beneficiary, whoever's going to be receiving it. And so from a, you know, for a very simple kind of viewpoint for human specimen work, you can think of the participant as the grantor. They're giving you permission and they're giving you part of their body, right? And the IRB of the ethics committee then is responsible for making sure that that is protected and that it is being done in a way that values their participation. And then the beneficiary is the researcher, right? They are the person who is charged with making sure that they do the right thing with the specimen. And just because they get the specimen doesn't mean they can do whatever the heck they want with it. I mean, the number of arguments I have had with individuals about, you know, when I asked, so what research are you going to do with it? And they're like, why do you need to know? And I'm like, because they agreed to these things and not these things. And I need to make sure that you know that, you know. The sort of biggest example of this is I did a lot of HIV research for a long time. And there were people who wanted to dive into those specimens and look at hepatitis C. And I said, yeah, nobody gave permission for that. And if you go in and make a diagnosis of hepatitis C, I'm now legally obligated to go back and tell that person because that has implications for their health, you know, and there's treatment and there's just all sorts of cascading things. And after explaining it about 15 times, I could finally see the light go on and the researcher got it, but it was really painful. Now, the truth is this can happen in multiple layers, right? If you're a biorepository director, you may actually have you may be receiving specimens from the initial ethics process, but then you're holding on to it and you still need to have a process for that release. So every time we release something from our biorepository, we do a check to make sure that everything is in place. We do not have an inventory that says, yes, they've agreed. No, we go back every time to say, did they agree? And does it still hold? And yes, that is a pain, but it makes sure that we're doing the right thing. And again, it gets back to do you have enough people to actually help you do all of this? When you're talking about animal specimens, there are other rules in place, right? So if you're collecting samples from animals, you know, usually IACUCs don't care once the sample is out of the animal, but there may be regulatory bodies, right? There may be laws or agreements, you know, CITES. We have to deal with, you know, fish and wildlife and agriculture, because these things could have implications for agriculture in the US. And I'm sure you have very similar regulations in every other country. So really, I think, when you're thinking about a virtual biorepository, these are some key considerations. There's the biosecurity aspect of it, which is who has access to see the repository. Is every specimen at the same level, or do you need to have a gradation, right? If you're seeing serum samples, that's going to be different than infected blood samples. That's going to be different than highly purified virus, right? And so think about that in how you're setting up the viewing rights. Do you limit those viewing rights? That may be something important to consider. Infrastructure, like how do we actually support this? Because this is not cheap. This is ridiculously expensive. And especially when you start, so I visited KEMRI in Nairobi, and I saw SMRF, and SMRF is great. And so I started asking some very specific questions like backup generators, and all of these things, because they've got a ton of -80 freezers, but do they have all the other infrastructure? And they do, and it's great. But not every place has that. So, it is definitely something to think about. And finally, the politics, right? So the sad thing is, when you're doing biorepository work, you have to deal with your institution. Occasionally, your local government may have a hand in it. Certainly, your state or provincial government has a hand in it. Your national government has a hand in it. And then international law applies. And you've got to deal with all of those to be doing the right thing. And so how do you navigate that? How do you set up the procedures so that you can actually do all of that? So that was a super fast overview of biorepositories and setting them up. I'm happy to share those slides, and I'm happy to take questions and talk about anything. I guess we've kind of run over a little bit, but.
Like the rhetorical question is, why would anyone want to do a biorepository?
Yeah.
What you said, four is the only guy in the room kind of thing.
Yeah.
Why would you want to do it?
I'll be honest, I never set out to be a biobanker. I did this by accident. I got a call one day from somebody who said, yeah, I got a new program and I don't have enough effort. Would you be willing to take this over for me so that I can, give up my effort on this? And that was how I got started, was I took over something that was previously existing and much smaller. And it was that, and the other story that I tell is, so I was the chief medical officer for the Duke Human Vaccine Institute for a decade. And I became the chief medical officer, not because I was like, I gotta be the chief medical officer. I was every week having multiple meetings with clinical people and research people fighting with one another. And I was the only person that actually spoke both languages. So what would happen is I'd have people on the research side being, why can't I get a blood sample? Like, all I need is for them to go draw some blood. And the clinical people being like, there's a person on the other side of that, right? And they have a life, and we have a consent process, and there's all this stuff we have to do to actually get that blood for you. So we'll work on that, but you have to understand that there's a process here. And then, of course, the clinical person gets the blood and drops it off. And they're like, I dropped off this sample that I worked so hard to get. Why did they look so angry? And I was like, you realized you dropped it off at 4:30 on a Friday, right? And so you've now just completely messed up their weekend because you didn't plan for what their needs were. So it was literally just getting people to talk to each other. And so I backed into this. It was because I actually knew both sides of it. And then once I had a biorepository, I started thinking of it as a small business. And how do we grow in a way that supports what we're doing, but makes it useful for people? And so I learned all of these things on the fly. But honestly, I would tell anybody wanting to set up a biobank the same thing that I tell anybody wanting to go to grad school or med school, which is don't. Because the truth is, if me saying don't makes you not do it, you don't want it enough. You gotta want it more than that, right? You gotta want it more than me saying no. So, Warren.
Thanks, Tony. Very entertaining and insightful at the same time. So, because most of our activities is around test development, validation, evaluation, right? We focus on antigen RDTs and verification specific because it's the fastest thing to do, and there's so many manufacturers. What we often face, and it's a frustration for me, biobanking person, is they don't want frozen samples because they need 30 positive, 30 negatives for a verification of an antigen. Yeah. Right. Is there data out there? Do you have to just confirm concordancy?
Yeah, so it's an interesting, there are so many things wrapped up into that. And let me pick it apart. So the first thing I would say is that a lot of times developers have a very narrow view of what they want. And if you talk to them, you can kind of guide them into a space that's a little more realistic, right? And there may be really, really good reasons to lock down why you want specimens from a very specific area with a very specific set of characteristics. Most of the time, they probably don't do that. And you just sort of have to talk them through it. But, getting them to sort of pre-specify those things is really helpful. The issue is a lot of times they don't know until you're well down the road of collecting specimens, right? So I do think that the efforts that we've been talking about leveraging what people have already collected and also setting up, a limited subset of very well curated specimens can bring significant value to the program. For example, it would be lovely to be able to, when SARS-CoV-2 started, to be able to have, we have all of these specimens from 2018 before this virus ever was around. And now we can compare them to this. And we know that they actually came from the same populations, right? To be able to do something like that, that would have been a tremendous value back in, say, May of 2020, right? And we didn't really have all of that in place. But I think we can start thinking, what would we want that to look like? But the other thing is the technology keeps moving so fast. I mean, that's the other thing. I think it's wise to not focus necessarily on a specific technology, but to think about the characteristics. Because, for everything that we're doing right now, 10 years from now, it'll be totally different. Or it'll be done in a format that we've never really thought about before. But this is the challenge of doing all of this, right? And oftentimes the shift in technology highlights the utility of samples that may never have been thought of as being useful. So I've talked to Nay and Judith about this. One of the studies that I was involved in was we got plasma, serum and PBMC samples from CDC that were collected as part of the WITS cohort, right? Women and Infants Transmission Study. So this was a big study that was done in the United States looking at HIV transmission, positive moms to their infants. And it was done in the late 80s, early 90s, before antiretrovirals were available, and really before the 076 trial had happened, so AZT wasn't really common yet. We wanted to do a genetic study on those samples. Nobody in the late 80s, early 90s was consented for anything like a genetic study because it didn't exist yet, right? So the initial answer from CDC ethics was no. And in fact, you have those samples, you should destroy them. And we said, time out. wait. Let's talk about this. And we went through a two-year negotiation process where we went through all of the ethical discussions around the use of those specimens. If there are critical questions we can ask with these specimens that are fundamentally unanswerable with any other cohort. You could not do that study now, right? You could not have women randomized to no treatment because we know that's bad. So we can't do that study anymore. Plus, the fact that they didn't consent for genetic testing, it didn't exist. So it's a little sort of silly to expect this. And then we went through the whole discussion of could we track all of these people down? And the reality is most of them were probably deceased at this point. So anyway, after 2 years of discussion, we did finally get permission from all of the ethics bodies, and we were able to do a study, and it was really, really informative. But yeah, this is what gets back to, like, do you have the staff to do this? Do you have the staff to pay people for 2 years to do these negotiations, right? I mean, it wasn't like 2 straight years of we didn't do anything else, but it was a lot of meetings and a lot of conversations. And, getting people to think about it in a different way rather than just the immediate no. Yeah.
I have a question for you, and going back to the biorepository being sort of a business. Do you have a retention policy? Do you have upfront sort of a decision on how long some samples will be kept or culling?
We do try to get all of that information as much as we can. I mean, it's simple for the studies that we're doing where we're holding them temporarily. So there we just say, we're going to hold them, we're going to send them, it'll be somebody else's problem, which is a cop-out, but it is what it is. For the Chavi repository, which is the bulk of my specimens, those are tied to a specific grant or to successions of that specific grant. And as long as that funding stream continues, we have permission. We renew that every time the grant is renewed. But we also know that when we get to the point that that stops, then we will enter into a negotiation with the NIH about what now, right? And so when we have studies like that, that's what we try to think through is how are we going to do that? For every study that we are doing now, anything that goes into the repository, we have a separate repository consent. Nobody is required to sign that. Any specimen that doesn't come from someone who signed that consent is flagged so that they're discarded at the end of the study. But if they have agreed to the biorepository consent, which basically says, we will use your specimens for as yet undetermined research, and that it will require ethics approval, all of those kinds of things, but that we're not promising we're going to give you any information back, and we make that very clear up front, then we feel comfortable about having those in the repository. But we also recognize that the laws can change like that, and we may have to revisit it all. It's part of why we go through this process before every release of making sure that everything's in place, rather than trying to have it in the database, because the database will be wrong. Yeah, but it is really important to think about those things up front. I completely agree. It's also important to think about it just because physical retention of that many specimens becomes burdensome at some point. And do you need to create space for something new, right? You can't keep building houses on top of houses. Eventually you have to knock some down to put a new house up. It's the same thing. So eventually I may have to empty a freezer for that reason. But yeah, we try to think about that up front.
Extension of sample storage at the biorepository, and yet the code probably it's stored somewhere else, like at the site. And according to their TMF, they'll destroy the records after a while. You still go back.
Yeah, that's so. We try. We do everything that we can to make sure that we have a copy of the record for everything that we have in the repository. Having it electronic makes life a lot easier for obvious reasons. If we are in a situation where we cannot find those forms, then our default is they can't be used. And I actually went through this just recently with some specimens that were collected in 2008. They were collected in the UK. And yeah, all the original forms had been destroyed. So we had to go through this process, and we've actually physically moved our offices twice since then. And, it's like any house move, things get lost. So it took time. We were able eventually to piece it all together to show that, yes, we had permission to do all of the things, but it extended our process by about 3 months to get the approvals because we had exactly that issue. But yeah, then you get into a question of how often do you audit those records? Because if you had the ability to audit the records and say we no longer have information on these samples, then we can discard those samples. But is that worth your time? It gets into a balance, right? every vial that I store costs a fraction of a penny per day to store. But when you're talking about 1 million specimens in a year, it adds up. So it's when does it become cost effective to have someone doing that work versus you just hang on to them for a while and deal with it down the road. But, there's no right answer for it. It gets easier the more you have all of this in an electronic format that's easily queryable, but yeah, if it's paper records, it's painful.
Yeah. Hi, everyone. We have a question from the chat on Zoom. It is, what can be done to ensure that researchers are actually using samples for what they have been collected? You say you check if consent was given, et cetera, and go as far as reconsenting. Clearly, you are a very ethical person. Sadly, not everyone is.
Oh, wow. That is a challenge. You are absolutely correct. That is a challenge. There are a number of protections in place now to prevent people from doing things unethically with their samples. But at some point, it comes down to trust, right? You have to trust that the individual to whom you were giving the specimen is going to do what they told you they were going to do. And unless you actually sit in their laboratory and watch what they do, you may not be able to verify that. So at some point, you do have to trust that people are behaving appropriately, right? The things that are in place now that are helpful in this regard is that mostly to get things published and to get anything through the US FDA, you have to have very explicit documentation about everything that was given in terms of permission. So if someone does something that is not permitted by the regulations, they won't actually be able to publish or to get a drug approved, or in some cases, even to get the patent approved. So that's something that's actually changed in the last 20 years, that those things have gotten a lot more stringent. Are there ways around that? Absolutely. And are there journals that aren't doing that? By all means. But for the most part now, you do have to document all of that. Now, could someone lie there? Absolutely. And are there scientists that lie? Sadly, yes. Yes, Judith.
There are in the US, at least there are legal precedents for people who are misusing samples. So there is more than just being unethical. You're actually doing something illegal.
Yeah.
And
Yeah.
so, okay.
Yes. Yeah, that is a big point, which is if you are shown to be in violation of those laws, you can have your grants stripped. You can be barred from applying for future grants. Largely, institutions would fire you. You would become persona non grata. But yeah, I mean, at some point you have to trust that people are doing what they say they're going to do.
Thank you, Tony. So I have one question, and going back to a previous subject, is that I worry a lot about samples and the separation of their metadata.
Mm-hmm.
And, we talk about it a lot, but we sort of wring our hands and we don't know what to do.
Yeah.
But are there on the horizon mechanisms in which the sample and the data can be connected forever?
It's a good question. And I think this is something that we as a community will need to work toward. But in some ways, it gets back to the earlier discussions around the difference in ethics regulations in each of the countries, right? Because, if dates in the US are considered PHI, sharing that is a problem. That isn't true for most other parts of the world. And so do we build the system that's going to do this to US laws or to EU or, South Africa, Kenya, wherever? Or do we try to take the most restrictive set of all of that which frankly, I'm not sure I want to be in that straitjacket. Yeah, I don't know that there's we don't have an answer for it yet. I think we need to work toward that. I think that actually would be an excellent goal of this working group that we've got going, because that's the key issue. I agree with you. Now, just, from a simplistic view, I'm a big believer in a barcode on a vial, and that barcode ties to the database. And then that database, you can use that to track all of the information. If you can put all this stuff electronically, there are ways of tying all of these things together. And I have very strong feelings about check digits and all sorts of other things related to sample identification that we could get into if you want. But yeah, I think there are electronic ways of doing this, but I think the regulatory landscape is actually the harder part of it all, because of all, I have to deal with HIPAA. I have no choice. I'm at Duke. So, and I suspect you have similar issues. So, yeah.
I just wanted to confirm how, especially this would benefit the participant.
Yeah.
If the samples come to the biorepository or the biobank, and they have no control.
Yeah.
And then you get what do you call them? The commercial developers.
Yeah.
You come and do research with the samples and get good results.
Yeah.
How will you connect the participants to get the benefit?
It's a great question. And you have a direct comment on that, or? Because I think there are really good ethical arguments for participation, but it is a challenge.
Thank you for your question. That is the reason why it's very good to include ethics. What is considered in such a scenario is the social value. So the IRBs are supposed to come into place. They're the ones who are supposed to oversee that in this particular sense, that at least when there's a benefit, it should be able to benefit the society from where the samples came from. It might not have an individual benefit, but if the samples came from, for example, Nairobi or Kenya should be able to directly benefit. And if that is not going to happen, that IRB should reconsider its approval.
And I'll expand on that. I do think that you raise a really important point, right? Somebody's going to benefit commercially from this. Yeah. And is there a mechanism to bring that value back to the community? Probably not in terms of paying participants. Participants get compensated for their time in participation in trials, but there's always a balance between coercive payments, which encourage people to give specimens even if they don't really want to, but I need the money and that kind of thing, versus saying, well, nobody should be compensated, in which case, why the heck would anybody participate in a clinical trial to begin with, right? So it's always a balance there. But I do think that getting commitments from companies who are developing these tests, who are then going to commit to, yes, we will then make sure these tests get deployed in the local environment to bring value to the community. And the reason that we need those specimens is we need to know what the test characteristic is in that population, right? So I can think of two big examples where not having the right controls makes a difference. I'm a pediatrician. I do pediatric infectious diseases. I used to spend a lot of time in the emergency room, and I'm from the American South, so there's a relatively large African-American population. And if you look at white blood cell counts between white Americans and African Americans, there's a difference in normal ranges, right? And if you don't know that, and you're simply basing it on one set of values, people look abnormal when they're not, right? The second one, which is actually more entertaining, how many of you have used an automatic faucet, like at an airport or whatever? Has anybody ever had problem going up and the thing not turning on? Happens more commonly to people with dark skin. It's because they were all calibrated to people that look like me, right? They didn't have a big enough test population, right? That's where the problem is. And especially if you're talking about Africa, genetic diversity in Africa, within the continent, especially south of the Sahara, is bigger than the diversity of all of the rest of humanity. So we need to make sure that we're representing that diversity when we're doing this testing, because we want to make sure the tests are valuable to you. It would be terrible to deploy, we do a study in Sweden and we say, there's the normal population, and we bring it here and it doesn't behave properly. That would be a terrible outcome. So, I think there are good arguments for having participation, but there needs to be a commitment. There absolutely needs to be a commitment to bring it back. And ideally to engage scientists who are here to be involved in the development, right? Back to the previous discussions about, we're no longer in a period where we need to take the specimens somewhere else. We need to bring the tests here. That's what we need to do, because we need to get people here working on the problem. A lot of smart people here. We need more smart people on the problem. I believe that wholeheartedly.